For those who don't know who I am, my name's Greg Porta. I am the Director of Advocacy and one of the original founders of the Child Foundation. A few years ago, I stepped back because I decided that what was more important to me was advocacy and research. And I turned the reins over to Ann, and she's done a fantastic job. She asked me to give a short talk, but she forgot who she was handing the microphone to. As the poor lady on my flight here for 4.5 hours was not smart enough to know not to ask me what I was heading to San Diego for. But she now knows everything there is to know about child. They asked me to give a short talk on advocacy and research, and a lot of the doctors right before me hit on some really good points. Dr. Cotton made a great point about how the senators and congressmen don't want to listen to the researchers, they listen to us. So that's what I do. I am a member of the ATS Public Advisory Roundtable. That is a patient group of rare diseases that advocate on the Hill for rare diseases and research. So what I wanted to do today is just to add a little bit to what all they were talking about. And I want to give you a few figures. $3 billion, $680 million, $160,000, and $500. And the way that I'm gonna tie those all together is, how many of the parents and caregivers know what the NIH is? It's the National Institute of Health. They fund over 80% of all the research that goes on at all of our doctors' institutes through grants. Their budget this year is $3 billion. Their budget last year was $3 billion. Now that may sound like a lot of money, but that's not a good thing. Because they are keeping the NIH level at their funding from last year, there's going to be a lot of good research out there that does not get started. Now, they also, unfortunately, on the deals that Congress and the House made, I'm sure a lot of you heard or went past and saw the deals that they made, unfortunately, are requiring major cuts. They're cutting the CDC, they're cutting the VA, they're cutting, and when I say cut, NIH, I feel like they're cutting when they're keeping it at the same level. We have to advocate for these doctors. We have to call our senators, we have to call our congressmen and tell them that an even keel in NIH funding is not good. We need more. They're actually meeting this week. That's where I came from before I came here. They're meeting this week to come up with that preliminary budget number. And that's what it looks like. They are cutting our research dollars. Now, the $680 million is the budget for one of our doctors spoke, NHLBI, the National Heart, Lung, and Blood Institute. That's who I advocate to. That's who I speak to on a regular basis. They have $680 million budget this year, and they are using it. Unfortunately, their greatest stepchild is the heart. So all their funding, they want to tout their funding for heart disease, and they want to tout the fact that the heart disease deaths are going down, although they are still the #1 cause of death in the United States today. But what they don't want to look at, and what we are here to push into them and make them understand, is that lung disease deaths are rising and the heart disease deaths are going down. So that's another advocacy thing that we can do by talking to our congressmen. The only people who can direct NIH to where they spend their money is Congress. That's why I spend a lot of my time up on the Hill trying to get them, trying to get resolutions for us to, so that they recognize child. Once we can be recognized as a disease by the Congress, then we can start asking them to start directing NIH to start funding more lung research. My next number, the $160,000, happens to be what we as a foundation have put forward for research. As Dr. Cotton spoke earlier, our very first research grant went to him and his colleagues. We felt that that was a very, very important subject. And it's something that needed to be looked into. We are now offering our second one this year. And let me step back, because that's my other function on ATS PAR. Through ATS, they are matching our money. Whatever money we give, they are matching it. So right now, obviously with the crunch and the recession and everything, things have kind of slipped back a little bit. But as long as they're matching our funds, they're doing all the work for us. They're sending out LOI requests for our grants. They're allowing our scientific board to review those LOIs and give their opinions. So our advocacy here at this low level as parents and caregivers is very important. My last number was $500, and I think one of our illustrious doctors spoke earlier that through ATS PAR, we fund a young resident as a travel award, $500. That $500 gets us somebody to the table. We bring in young residents to ATS to present abstracts, just like they had the abstracts here this weekend. We bring them in from all over the world. And just for $500, we're bringing them to the table, and then we grab a hold of them, and they're beholden to us. So I guess really to wrap it all up, what I'm trying to say is our advocacy, as small as it is, gets us so far. The $160,000 grant of money that we've put out in the last 2 years, I see bringing back tenfold. Just an example, ATS PAR in the last 8 years have put out approximately, they've teamed up approximately $10 to $15 million worth of grants. And in that last I'm going to say 8 years, that has spawned $98 million more in research funding for those grants that were issued. So what I'm trying to say is that $80,000, the first grant that we gave, and the $80,000 we gave this year, in 10 years is going to be tenfold, because those doctors, those young residents have started their research. We gave them the basis to start it. Now they're going to be able to go to the NIH, the CDC, the VA, DOD, and say, I've got this base started. And they're going to be the ones that are going to be up the front line for all the other grants. So please, I can't tell you, this is a very passionate subject for me. And it's one of the reasons why we started this. So I'm sorry. I apologize. Please, you are your best advocates. Call your senators, call your congressmen, and tell them we need research. So with that, again, I apologize, but I was glad that Ann asked me to be the moderator for this session. because these two gentlemen that are going to talk to you now, I believe, is where a lot of our research, a lot of our breakthroughs are going to come through, is through genetics, gene sequencing, and I won't bore you with all that. But our first speaker here today for this session is Dr. Larry Nogi, Division of Neonatology, Johns Hopkins University School of Medicine. And he will be followed up with Aaron Honvis, Division of Newborn Medicine, the St. Louis Children's Hospital, Washington University.
Thanks, Greg. It's a little hard to follow, but I'll reinforce what he said and what Dr. Cotton said. The congressman, the NIH isn't going to listen to us, but they will listen to you. And so the power of grouping together and advocating is far more than sort of our individual voices. I want to also say how happy I am to be here and to be meeting and seeing some of you. Some of you I know from the ether and email and voices, and it's actually nice to sort of see the faces. What I'm going to talk about is a little sort of overview about genetics, and I'm going to try not to get too mired in the details, because frankly, we lose our students and residents and physicians who call me on the phone when we talk about this. But for Aaron and I, this actually started with one patient and one family some 20 years ago. I was off in my laboratory working with cells and with rats, and I literally got sucked in with one child that was sort of a mystery, and that sort of started this sort of whole foray. But I want to start off with a little sort of philosophy of disease. And this isn't just a theoretical construct. This is actually how we've reformulated our curriculum for our medical students in terms of how we think about illness and how we think about wellness. And it's really that it's all genetic. Well, it's really that it's all environmental. Well, the point is it's both, okay? And rather than thinking about illness or wellness as something bad luck that happens, so why did this happen to this person, trying to think about what the combination of genetic and environmental factors are so that this person is manifesting this state of wellness, okay, I'm still going to have to come back to the keys, or this state of illness at this point in time. So what this is just a sort of little seesaw, and I've got on the far left some genetic conditions or things that are going to primarily drive how well someone is, and I've got on the far right auto accidents, which is something most people don't think about as genetic, but we are learning that there are behavioral conditions which are genetically imprinted, so that risk-taking behavior, coordination, attention, so that they all sort of factor in. And then most things are sort of in the middle, in which diabetes, cancer, emphysema, there's a combination of genetic and environmental factors, and how can we manipulate both so that we can drive people towards wellness as away from illness? So why is it important to find genes? Okay, and there are a couple of reasons for it, and I'll go through, and what I've tried to emphasize is where we've had some successes, where we are still working on things, and where we still have challenges to go. Well, first of all, it can provide an explanation for an illness and understand how and why disease occurs. And I think for a lot of you in this room, just sort of knowing that there's a name and an understanding can be helpful, as opposed to people sort of scratching their heads saying, well, we don't really know what this is, we don't really know what causes it, but can we at least begin to sort of understand? Secondly, it lets us begin to classify illnesses based upon causes rather than just a grouping of symptoms and findings. And that is the first step to trying to figure out how things happen, how things go awry, and therefore how can we devise therapies that are based on what the underlying cause is, as opposed to just throwing things at it because we think it might work, and maybe it will, maybe it won't, but there are lots of side effects. So the analogy is, how can we go from throwing large-scale bomb set problems to designing smart missiles that are going to actually get to the problem. A third is that we really want to improve our diagnostics. We want to be able to diagnose someone's condition through, if we can, a simple blood test or a simple spit test, rather than to have to go through a whole battery of things that have their own side effects. Radiation for CTs, anesthesia, and cutting someone open for lung biopsies. So that's where we'd like to go, and we've achieved some successes. We have some illnesses now that we can diagnose non-invasively, although there are still issues related to interpretation of testing and cost, so we're not there yet. As far as genetics goes, it's also important for accurate counseling. We want to know that other family members, if they're at risk, what's the chance that this is going to happen again? Can we prognosticate what the disease course is and give you information about what to expect? And then lastly is that we want to eventually translate this into something that we can use to help the people with the illness and the children. And this is where we still, unfortunately, have a long way to go and what one of the challenges is. So let's just do a little sort of primer on genetics again. And what is a gene? Well, it's just an inherited unit of information. And I think of it as a blueprint or set of instructions, okay? And it's instructions for assembling a protein. And genes are made of DNA. DNA is actually a fairly boring molecule. It's a very long string of one of 4 bases, A, C, G, or T, that are just strung together. And the genes are organized into chromosomes. And these are 23 discrete groups of DNA, and then there's a much higher organization, and you've heard about epigenetics, because how the genes are packaged into the chromosomes matters in terms of how things are read. There are 3 billion bases in the human genome. That is a large number. That's 3 billion individual pieces of information. Every time a cell divides, this is copied over, okay? If we were to play the old game of sort of telephone and start a sentence going around the room, or just to write a sentence down and pass it on, of only 100 letters or so, the odds are if we passed it around the room, by the time it got to the end, there'd be a mistake in there. So it's pretty easy to understand when a cell has to copy over 3 billion pieces of information each time a cell divides, that things can go wrong in the machinery, and that's how changes occur, and that's how evolution occurs. There are some 20 to 30,000 genes in the human genome. This is actually a number that is a bit of a moving target. It seems to keep coming down in recent years. But the reality is, for those 20 to 30,000 genes, we only really know what a quarter of them do, is the current estimate. And there's still a lot that we don't know. And the other thing is that everyone has some genes that don't have the correct instructions. And that's, I've got the some genes in, what color is that? Green. Okay, I would have said red, because my particular genetic disease is that I am red-green colorblind. And I actually can't tell you what color that is, whether it's red or green. I actually did that deliberately. It's not a huge morbidity to me. I can't get a pilot's license. I get a lot of criticism from my wife and daughter. It's like, why did you wear that combination of things? But fortunately, it doesn't have a major impact on my day-to-day existence, but it is a genetic disease, and it just happens to be the one that I know that I happen to have from very early on in childhood. But the point is that all of us have things that differ. All of us have something that doesn't do what it's supposed to do. So how is DNA organized? It's the 2 strands that run in opposite directions. It's twisted into this double helix. And each strand is actually the complement of one another. They're sort of mirror images, if you will. And this is how it's able to be copied over. And it's the sequence of these letters, these G's, C's, A's, and T's, which encodes the information. And it's sort of grouped into 3s, and those are what are called codons. And then the other important thing is a large percentage of DNA doesn't directly encode information. This very commonly gets called the junk DNA. You'll hear that sort of on the news or in reports. It's really not junk. It's called junk because we are not smart enough to know what it's doing. But it's clearly there for a reason. And I think as scientists and physicians, we believe that. But it's a problem because it's an awful lot of information there, and there's important things that reside in there, and it's actually 99% of the genome. So when we do a lot of these genetic studies, we're looking at very small percentages, but we're missing things a lot of the time because there's important information in the areas that we're not looking at. And we don't look at it both for technical reasons, for cost reasons, but largely because if we start to look there, we're going to find things but not know what they mean. So again, some more definitions. I've talked about what a gene is. It's the section of the DNA that tells the body how to make a single protein. The proteins are the basic building blocks and the machinery in the cell in the body. And in between there's something called an mRNA. That's what sort of the gene is copied into. Parts are whittled down to the essential parts, and that's what's then translated into making the protein. And then lastly, we talk about the phenotype, which is the physical expression of the genetic material and all the environmental influences. So what can go wrong? All right. So, well, all or part of a gene can be missing or duplicated. I'm going to step back before I trip over something. Okay, a piece of a gene can be physically turned around and can be in the wrong orientation. There can be a small piece that's missing or inserted. There can be a change in a single base, and this is one of the most common. And the terminology here gets confusing, and there are scientists who struggle with this. So a mutation simply means a change from what we think the usual sequence is, and it gets used interchangeably with the term sequence variant. But the common parlance that most people think of when they hear mutation is that this is something that's bad. But really all it means is that it's a change. And then a polymorphism just means it's a change that's found in greater than 1% of a certain pool that one looks at. So how do we find genes for illness? Well, historically there have been two major approaches that scientists have taken. The first is what's called a candidate gene approach. So what we do is we start with a gene or a protein, okay, and try and see if it has a role in an illness. And how do we pick that gene or that protein? Well, it's really an inspired guess. And we make that guess based on what we think that gene or protein is doing in normal physiology. Or sometimes we have clues, because people such as Jeff Whitsitt have been playing with genes in mice, and they knock a gene out, and they find out that when they knock the gene out, the mouse has characteristics that look like a human illness that we see. So an example for that, for example, is alveolar proteinosis, which is one of the forms of interstitial lung disease. And about 20 years ago, some scientists knocked out the gene for a gene called granulocyte macrophage colony stimulating factor. They were interested in this because they thought it had something to do with the blood. Well, the mice's blood was perfectly fine, but then they got this funny lung disease. And that's led into insights into the cause of this rare form of lung disease called alveolar proteinosis. So that's sort of a candidate gene approach. The other is what's called linkage analysis, and that's where we start with an illness of interest, and we try to find the causative gene by studying families in which the illness occurs. And then we try and figure out, by sort of looking at what are called markers, that sort of just little tags, if you will, on a map, okay, in the human genome, what goes with the disease and what doesn't, and then sort of working backwards. And that's how the gene, for example, for cystic fibrosis was found, through sort of a linkage and a reverse genetic approach. So those are historically the ways that we have approached things. So what do we do when we find a gene? Well, we've got an explanation for an illness. It means that diagnostic testing is feasible, but there are some caveats, and these are still some of the challenges. The interpretation may not be straightforward. It's still expensive, and it's slow to get results. The good news is that the technology keeps improving, and things are actually turning around fairly fast, and the costs are going to keep coming down, and the time to which we get results is going to come down. We still don't have sufficient information for good prognostication. Genetics isn't everything. The environment's important. And increasingly, we're learning that genes don't act in isolation. They act together, and there are things we call modifier genes, and they interact. And probably most frustrating for us is that the translation into therapies is slow. So what are some of these new technologies that are coming along? Has anybody here seen the movie Gattaca? Okay. A movie I highly recommend, by the way. It sort of flew under the critics' radar screen, probably because it was a little plodding, not as much action and adventure and special effects, but a very thoughtful movie. There's a scene in there where Uma Thurman takes a hair from the character who's her love interest, Ethan Hawke, and goes to this little kiosk and hands the clerk the hair. And the clerk sort of runs it through a machine and comes back and gives her a whole stack of papers, which is his genetic blueprint, and hands it to him and says, "Oh, you've got a good one." Well, this is kind of where we're not too far from with what whole exome and whole genome sequence are. Whole exome sequencing refers to those parts that incorporate the coding information that relate to the proteins. So it's a fraction. It's about 1 to 1.5% of the genome, but it's the parts that we think are the key parts. The whole genome is just what it says. It's practically pretty much the whole genome. There are some parts that aren't in there because there are some technical issues, but that's looking at all 3 billion bases. Now, that's a lot of information. I mean, this has become a very cost-effective means. Whole exome sequencing is around $1,500 on a research basis. Whole genome is $25,000 to $3,000. Well, that's not cheap. I mean, an individual genetic test can easily be $1,000 to $2,000. So this can potentially get us right to the variants in genes of interest, but the problem is it's almost too much information. What we're finding is that we are all very different, and trying to sort out what those differences mean is really the challenge. And I'm going to turn it over to Aaron, who's going to go through an example of this approach. But this is sort of where things are moving, and we're all going to be decoded soon enough.
Thanks, Larry, and it's a pleasure to be here. So I'm going to walk through a couple of the issues with whole exome sequencing, because we've had some, at Washington University, we have the good fortune to have one of the Human Genome Project centers on our campus, and we've interacted with the geneticists there. And so we've really been able to utilize that relationship to really start to make forwards and utilize these new technologies that are going to become even more pervasive. So actually I have a mistake here. It should be 1% of the genome, as Larry said. The exome is the entire protein coding sequence of the genome. So it's about 1% of the genome, as Larry said, and it's about 30 million base pairs. So we're looking at a small fraction, but it's still 30 million nucleotides that we're looking at. Now, when you get 30 million pieces of information, how do you really deal with that? I know when I'm dealing with large data sets, once I get beyond about 5 or 6 pieces of information, it starts to break down. And so multiply that several million times, and you start to get into some difficulties. And so the approaches that have been taken so far to try to get down to single genes or a few candidate genes have utilized a lot of assumptions in trying to take this 30 million pieces of information and get it into something manageable. And so the first assumption that's made is that whatever the cause of the disease is, or whatever the disease you're looking at, is caused by a defect in a single gene. Now, for many types of diseases, that's probably an adequate assumption, but I think that based on some of the preliminary data from exome sequencing, in which investigators have gone through hundreds of individuals who've had their exome sequenced, the current thought is that only about 20 to 25% of the time are you going to come down to that genetic sequence or the gene that's associated with that particular phenotype or disease. And I think that shows, number one, is that sometimes it may not be a single gene defect. Sometimes it may be a defect or multiple defects in those non-coding or that junk DNA. Or it may be epigenetic and have nothing to do with genes at all. And so even though we have the capability of doing the exome sequencing, we can do it pretty quickly too. Whole exomes can be turned around in about a 2-week period of time, at least getting the sequence data. So that first assumption may limit our ability to find things. Right now we're limited by our informatics capabilities as well. So I like to tell people that, right now the sequencing part is the easy part. The tough part is taking all of that information and then utilizing it in some real fashion. The other a couple of other assumptions is, number one, is that the disease or phenotype that you're looking at is inherited, and again that gets into the epigenetic portion, the interactions of the genes with environment that may or may not be present. And then a lot of times, since we're dealing with rare diseases, the other assumption that's made is that the genetic variant is going to be rare. So as Larry said, we all have lots and lots of variants in our genome. Some of them are more common. Some of they're variants where 40 or 50% of the population might carry them. There are some variants where less than 1/100th of 1% of the population carries them. But since we're typically dealing with rare diseases because of natural selection and evolution, we make the assumption that the gene of interest and the mutation or the variants of interest are going to be rare in the general population. So those are some of the assumptions that may or may not be hold true at this point. And like I said, right now there's about a 20 to 25% success rate in finding a gene. Depending on where you go, we're talking about $1,500, $2,000 per exome. These costs are decreasing dramatically, and one of the holy grails has been the $1,000 genome, and I think we'll be there within the next 1 to 2 years. So this is happening very quickly. The time involved, so when you think about the Human Genome Project, took about 10 years to decode all 3 billion sequences. We're getting to the point where we can do a whole exome in a few weeks, a whole genome probably in about a month or so. Again, the technology is changing, and we'll be able to turn around these genomes in a day. So the technology is changing very, very rapidly. So let me just go walk through one of our success stories. I don't think this is an interesting patient. I don't think that she's got a child disorder, but she had enough kind of other unusual phenotypic types of things. But I really wanted to illustrate this just to kind of give you an idea of the scale of kinds of things that we're looking at. So this was a little girl who actually presented in our neonatal intensive care unit with multiple problems.